And thank you so much. So, very good evening, good morning to all the participants, and I welcome you all to the machine learning based modeling of drug properties live webinar session. I am Divya here from Altem Technologies, Senior Application Scientist for BioVR Discovery Studio Support. And you all know that, as I say, the machine learning is the most common technique, and it is booming across world. Moreover, the technology of the integration of machine learning, it's gaining a more importance in the various domains. However, it is not limited in a pharmacological also. That's why today I'm uh, going for an introduce. I could say that we are going to explore the importance and applications of machine learning based modeling and drug properties. Let's walk along with me and then see how we can apply this technology and to improve the drug properties and to prevent the failures in a farm, prevent the failures in of the drugs in your post clinical phases and all. Let me share my screen. Okay. So machine learning based modeling, as I said, we are going to see how this machine learning algorithms can be utilized to understand the properties of the drugs. If you know the properties of the drug, say for an example, it is going to make a toxicity, it is going to bring immunogenicity, or it is having certain kind of side effects or hepatotoxicity, certain kinds of properties which is not required for the drugs, and there are certain properties or required for the drugs. Say for instance, you want to predict a number of hydrogen acceptors, donors, surface area. So all these things are need to be investigated when we go for a drug designing and development. So today's session is all about how we can model these kinds of properties using machine learning algorithms. Let's walk along with me. I was about to introduce my company. Altem Technologies is one of the leading company in 3D printing technology, and one of the wings in life sciences is a BioVR Discovery Studio, Material Studio, and then Bio 3D printing. And when I explain more about we must know about our BioVR, because BioVR Discovery Studio and the pipeline pilots or the two softwares have huge impact and roles in the modeling of machine learning algorithms. So unified lab management, process production operations, quality and regulatory management. Of course, whatever the data we try to extract, we will try to compile our data and process the data and document the data, and then we move to the quality and control management to analyze the data. And after that, only it will be integrated as a part of the tool, and we go for validation and testing, and it will be utilized for the users. So this is how we'll be exclusively works on quality and regulatory management and more into the collaborative science. When I say about a collaborative science, if you have a sequence, say for instance, I have a sequence, I don't know what is a drug target that I'm going to focus on. So if I have, say for instance, on the other hand, I have a drug targets, or I could say that I have certain compounds in me. I don't know where I'm going to end up. So either stage, maybe you have a compound or you have a drug target. In between, how to perform this process is the most important in your collaborative science. So if you have a sequence, it will model automatically and you can predict it, and then you can identify the site. Then you can see the interactions between your drug and ligand complexes. Similarly, if you have a compound, either you can go for pharmacophore based drug designing or the ligand based drug designing or the QSA approaches to understand the structural nature, and accordingly you can choose in a specific receptor assay and then target with the protein to seeds interactions. So either way, one can join in this predictive models, or I could say that Any of the inputs can be given and can be utilized for your analysis. And more into novel therapeutics. So we all know that novel therapeutics is a biologics. So if you want to develop an immunoglobulins or engineered ways, or I could say that different kinds of monoclonal antibodies, something like that, big molecules or large molecules or biologics. We need a certain protocols and technology to do that. So we have a specialized immunological based tools, and for model of antibody also we have a specific tools, because modeling of normal proteins is quite different from a modeling of antibodies. So we have that tools, and we even you can predict the immunogenicity, and also you can see the aggregation scores, and what are the properties of pertaining to that, that you can able to analyze it. And then if you sell about, if you identify certain drugs, and definitely it should be delivered. Say for instance the disease is like a cancer, delivering of a drug is a it's considered as a daunting task. Why it's so? Because cancer is not a single protein involved process. The network of proteins are playing a major role. So delivering within a specific drug vehicles like a nanoparticles based drug delivery, or a polymer based drug delivery, or a carbon nanotube based drug delivery, all are integrated as a part of the material studio software. So one can construct the polymers, and you can study about its behavior and its actions, and what could be a thermostability parameters, what is a physiochemical parameters. All these parameters you can analyze, and you can come up to draw the conclusion. And next, if you move into we have into chemicals and formulations. When you say chemicals or formulations, say for instance, if you want to develop a certain kind of a products, maybe a product for an, maybe a, I could say the agricultural product, or it may be a product for an beauty products, whatever the products that you want to develop, make sure, what will be make sure we shouldn't have any irritancy, or it should not cause any carcinogenicity or mutagenicity. So like this, we will try to analyze an n number of parameters. And also, if we get in a set dosage ranges, say for instance, this is a percentage that we need to treat this particular disease. If we know all these things, that and all, it's a boom for us people who is involved in this kind of research. So that's why we are having even the machine learning built algorithms which enhance these process in a very easier way. So let's see and walk along with me to know how we can predict these properties, and then moreover, what are the properties are more important for the drug development. So we are going to utilize the machine learning algorithms, one of the domain in artificial intelligence. So first, drug designing. So as we say that, so drug designing is not an easy process. Obviously, it will consume a more amount of time and then money and human intervention. So even though if you have been succeeding with your drugs and it comes to the market, there are certain failures are happening. Why it's so? Where the failure is occurring? Because of why? If you keep asking these questions, why, what happened, because of what parameter has been failed. So even though you tried very well in vitro assay, and then you draw the conclusion, so lots of queries will try to arise how to overcome this. That's why, in order to check the efficacy or the biological efficiency of this particular drugs, and also to prevent the failures, we are being depending upon the machine learning algorithms. Earlierly, we'll be trying to use only the docking technique, but of course, docking is also the one of the most prominent technology to understand the receptor-ligand interaction. But apart from that, if you have an now the trend is going towards a big data analysis. If you have a very big data that need to be analyzed and that need to be processed, so usually the tools like visualization tools or graphical interface tools are less compatible to handle more than millions of compounds and then data. That's why we are depending upon the machine learning algorithms. This, instead of screening 1 lakh of molecules at 1 hour, if we can able to resolve, if we can able to sort it out 1 million compounds in a 1 day, that is a great boom in our drug discovery process. So that's what we are going to see today. How ML will support in drug discovery and development. Say, for instance, In future, it is going to be precision medicine and the big data driven drug discovery process, as I stated earlier. Precision medicine, in the sense, I could say that in maybe there might have been future may occur like this. Even we are going to have our own DNA in a pen drive, and when we have certain diseases, instead of going there, you can send your pen drive and can check what of the diseases were being expressed, what are genes are getting expressed. If the genes are getting expressed, the doctor may prescribe you to down-regulate that specific genes so that it will be very specifically will be treating you with a certain kinds of diseases or the drugs. So that is called a kind of a precision medicine. So precision medicine is going to be a boom in your pharmaceutical domain. And how to introduce this. See, when we take the sequences and saying expression, how we understand, or if you have a big data in your hand, how do you take and prescribe the drugs to the patient? So it's all a very big task, but it can be done by using machine learning algorithms. And of course, drug repurposing. We all know that now in the current pandemic situations, we are undergoing a drug repurposing concept. People are trying to take drugs from various environments and testing against an SARS-CoV-2 to control the replications and to prevent on and also to reduce the infectious symptoms of the coronaviruses. And even if you say that supervised learning and unsupervised learning, these all are the different methods that is in machine learning algorithms that will help us to assist and direct us for drug discovery and development, and of course to study the properties also. And moreover, this artificial intelligence approaches in your ligand-based drug ritual screening in Discovery Studio will help you to screen a lakhs and lakhs of molecules under the various physiological conditions. Then, if you understand, you need to know the importance of machine learning and drug discovery. So the machine learning, I told you, it's a branch of artificial intelligence that based upon the idea of learning the data and identifying the patterns and making the decisions with a less human intervention. So as soon as if you get the data, we should make a decision that this is because of this gene expression, we need to treat with a certain kinds of drugs. So that kind of analysis or need to be integrated in day-to-day process to reduce the disease infectious. And moreover, this artificial intelligence in a drug discovery will help one to optimize the drug candidates using various combinations of ML methods. If I say ML methods, there are quite many methods out there actually. Which method we have to choose, how to choose, it's not a big task, but if you keep practicing, it will come to your hand. And moreover, the development of artificial intelligence will play a major role in screening a covariance in pharmacokinetic data. We have a pharmacokinetic, you want to study what is a adiabatic properties, that is a kinetic properties and dynamic properties like immunogenicity, carcinogenicity, and even the LD50 value. You want to predict all those things. For all those things, we can introduce various machine learning algorithms. And at last, it can be utilized often in a drug discovery than in development. So this is a crucial significance of machine learning in drug discovery process. Okay, how the scientist? So you people are scientists, right? How do you approach this machine learning algorithms? There are two ways actually. One is the pre-built approach. What is this pre-built approach? That means some machine learning specialist, okay, who is very having very strong knowledge in data sciences, may develop the models, okay, may develop the model, and that is applicable, that is available for the public or a private proprietary data sets like in Discovery Sphere Pipeline Pilot is available so that the scientist will deploy that model. So you don't need to know that if I say that a component, and if we come to the pipeline well, we call as a component. Component for TopCat, top component for ADMIT, which is inbuilt within a certain scripting languages and programs. So we are just take only that component, and the scientists, like you people, will apply this model to their problems. Whether my compound is having good absorption, distribution, metabolism, toxicity, like that, you can able to differentiate your compounds. Example, so if we say top cat models, is present in Discovery Studio, imaging crystallization model, and QSR workbench are quite common examples of pre-built model approaches that is a part of a BioVia Discovery Studio and then a pipeline pilot. And if I say pipeline pilot, you can do wonders in pipeline pilot actually, where you can edit your data and you can replicate your data, you can clone your data, and you can build up your own kind of a protocol. One can design a new protocol with that. And for using a pre-built model approach, you no need to require any understanding of machine learnings. You don't need to do no need to know that whether it is doing in the which algorithm it is using. Is it fuzzy algorithm or k-means or navy PSEs? So like there's a regression algorithm or they say there is lots of algorithms. So random forest. So no need to understand the machine learning what algorithms are using. But scientists can deploy that model just in terms of an applications. So this is we call as a pre-built model approach. The next one, it's a do-it-yourself approach. That means scientists or the engineers acquire the data. Now I have a certain data with me. I want to build the models from this data. And then they will apply this data to their own problems and deploy them to the other scientists. And that's why, for example, in 20 plus pipeline component learners are available here, and you can utilize and build the model. And of course, QSR workbench is also along with the protocol where you can use in DIY approach. But this approach requires some understanding of machine learning by a scientist. So these are the two approaches of machine learning for the scientist, which can be applicable in your drug discovery and development, and also to understand the properties of your drug molecules. Okay, now you people will be cracking your brain, right? So how to do this? How to fit for purpose approach? And what applications of this ML will guide me to answer all these questions? First question, what is a drug discovery and development needed by or you need to be utilized using machine learning? All right, moving on. What ML methods are most appropriate? So I know that I am going, ML methods are available, machine learning methods are available. What method will be appropriate to address my problem? Oh my God, again, there's again question. Which method I am going to use? And then what data? Again, it is a very huge bam, I could say that. What data that you are going to use to support this ML methods? When I think about it, the solution is very easy. I could say that you no need to crack your brain in deep when you want to understand ML. Basically, because why I'm telling you all, we already learned this ML methods in our past days. Only thing is we don't know the applications of that method. If I say, you will be getting that in your mind. Oh, all right, I read about this methods during my school days or college days so that it will be utilized. But I don't know the application. Now we are going to turn that your ideas into an applications. Let's see that. What is a drug day? Why it's required? What is required? See that in every single day we are getting 2.5 valentine bytes. Can you all imagine that? See, every single day, today, tomorrow, so data will be keep on replicating and multiplying like a matrices. Okay, we don't know where we are right now. To analyze this data, it is not an easy task. It is a daunting actually. We need a specific tool to do that, or certain programming to do that, so that you can come to the draw the conclusion. Moreover, it is not a quantity of data, but it is an opportunity actually, I could say that. It's an opportunity for all to generate a knowledge that matters the most. So where and all this data will be created? Say for instance, chemical structure data. Each chemist, organic chemist people, medicinal chemist people, pharmacological people, like this, there are lots of divisions that they people will try to create a various chemical structures. If one lab is creating 100 structures, How about 100 labs in Karnataka or going to create? Then what about in other states? So if we think about only the generating of chemical structures, that itself will end up with more than I could say the more than millions or 10 lakhs of compounds per day. How we will analyze this data? How do you understand which or a compound will be more efficient treating a disease? So all these things are keeps running in our mind. Then comes to the biologist. They are using a gene expression and genetic data. Okay, A gene is expressing, B is not expressing. They will try to use various technology called next generation sequencing technology, chip sequencing technology, and RNA sequencing technology, microRNA, bacterial sequencing. So gene expressions in humans, bacteria, fungi, virus. So it is again a very huge domain. Genes are getting expressed, which gene is expressing, which is not expressing in which patient. Okay, and what is the genetic information? Then comes the high throughput in vitro data, screening the compounds within different drugs. Clinical trial data, one compound when it enters into clinical trial that will try to create a piles of data, as I could say that. Then electronic medical records, publical data, commercial data, internal companies data, and data is participating among the institutions. So like this, it keeps on piling up. The data set keeps on piling up. That's why the big data driving discovery has been huge among the data scientists. So, how to handle this and how to enhance your research and accelerate your research? So, for all these things, we need an ML method. Machine learning algorithms are required. Okay. Where and all you can apply the artificial intelligence drug discovery and development. Okay. Some of the applications for first of all, you can understand the pathway and finding a drug targets. So you can know about if I say about the pathways and drug targets, you immediately you get a stitch compounds databases, okay, and then string databases for protein-protein interactions. So all these things or protocols or will help you to understand the pathways and finding the targets and finding the hit or lead. So how many hits are there? How many hits have been generated? Synthesizing of drug-like compounds. It's again a very huge task for the synthetic chemist. What is the mode of action of your drug? Agonist or antagonist or partial agonist or partial antagonist? That requires even still more knowledge. Drug repurposing. If I have taken certain drugs, how I am going to repurpose these drugs? Excuse me. Polypharmacology, selection of population for clinical trials. So for all these domains, we can apply our artificial intelligence. First of all, let's see about an understanding a pathway or finding a molecular targets. So target identification is to treat a disease. So they will be utilizing in a system biological approaches. System biological approaches includes both the genomics as well as the proteomics. And through that one can understand the potential targets. So see here you have a number of targets, maybe around 300 that will be utilized in a preclinical phase 1, phase 2, phase 3 like that and number of non-targets and that were being divided into the predicted and non-predicted and it falls in the stage of a suspended discontinued withdraw and genetic associated data. So where we'll be using animal models with the disease prevalent phenotype as a great predictive power to understand the novel therapeutics, especially gene associated based diseases. So predictors, say maybe a training set need a cross validation and they'll be getting a non-target, target, what is the sum, and test set, non-target, target. So anything is data that you're trying to get that will go for a cross validation and then we'll be evaluating your results. So in this way, you can understand the pathways or you can be able to identify the molecular targets. Next, identifying a hit or lead. That means it will be enumerating of fragments lots and lots of fragments. Through that, you can identify the target specific virtual molecules, especially in R&D expenditure. The molecules of synthesis is not an easy task because you must need you have to retain a maximum yield and in the shortest process. So what the people will try to do, they will try to, you can see here, First, they will see the functional groups, valencies, double bond. So, like that, they will try to develop the structures. So, this is called as an in silico technique for ritual screening or structure-based drug designing. So they will introduce a various rules. Say, for instance, I am taking a lakhs of molecules or millions of molecules. First screen with area maybe, next screen with top cat, next screen with lipids, crystal of fire, or drug likeness properties, next screen with a docking, next screen with a molecular dynamic assimilation, and come out with a drug or MMPBSA method, draw the drugs. And with the drugs, you finally go for in vitro assay. So, like that, identifying the hit and lead or can be you can be like made like a pipeline that will enhance your process. All these things requires a less human intervention, and then I could say that more amount of initially it needs only one required development. That's it, and afterwards goes in a hand. And synthesis of drug-like components. So if you have a medicine chemist who have been aware of this retosynthesis, reaction mechanisms, steric and non-steric effects, and also it will help you to predict the best south after the reactions by minimizing the scope of your compounds. So in artificial intelligence platforms, you can see the three ends and three ends. What is the three ends? Or nothing but a deep neural network with Monte Carlo search and cause. Okay, so computer aided organic compound synthesis and promising a building blocks for an selected and well-known reactions for the synthesis of target compounds. So, like this, you can introduce a synthesis of drug likeness compounds and then predicting the mode of actions. Say, for instance, what mode of action that is required? Maybe an absorption, aqueous solubility, blood-brain barrier, plasma protein binding, CTYD binding, hepatotoxicity. So these are the very, very important properties of your drugs, which need to be analyzed perfectly before going for a drug discovery and development. And moreover, top cut is also considered as an important parameter. And the next important property is a top cut. So when I say the drug properties that need to be modeled, these all are considered as some very, very crucial properties. How much absorption it is taking place, whether it is solubilizing or not. When your drug is going to obey this property perfectly and you are screening initially without going into the in vitro technique, that will definitely will save your time and improve the drug discovery process. Moreover, when you can understand even the fingerprints, that is also most important properties of drugs. Fingerprints are playing a major role. Placing a fingerprints at one position will have a good bioavailability. Placing a different position may cause a toxicity. So how do we understand all those things? It's not so easy, right? By an human can know that how many fingerprints he will copy for top cat prediction, how many he'll do for absorption, how many he'll do for solubility. So all these things are not an easy task. That's why we will be depending mostly into an machine learning kind of an algorithms. Okay. First of all, if I take a drug, I will anyhow we will see the demonstration of this area properties will help you to understand more about this of the drug compounds and its associated properties. So,